can be cured by a very low cost. But if we are curing the disease in later stage, we might need to pay 100 times more cost. So here, how we can use early disease diagnosis, and that we are planning, like in the case of tuberculosis, how it's spreading in the society. And if you know, the TB is usually the TB in the latent phase, and latent phase we cannot ever detect the tuberculosis. So here what we are planning to detect the TB in the latent phase, and as you can see that how the TB is increasing if you compare from the 99 to recent. So every year we are getting the increase of the population of tuberculosis person. And here I think we need to develop some low-cost device, because the sophisticated instrument cannot be delivered to everywhere. So here we need to develop some low-cost that can help. So in general, for tuberculosis, we use the PCR approach. And in PCR approach, the normal cost is very expensive. It cannot be affordable in every laboratories. Although we have such type of small PCR machines, but still the cost is a matter of. And so what we are planning here, because in general PCR there are some Peltier methods, and the Peltier method is very expensive. So how we can come up with new idea, like using a isothermal PCR, which is only a single temperature is needed. We don't need to have a 3 different temperatures. So using a single temperature, we can be able to detect the tuberculosis, and that's what we are approaching here. That we call the LAMP, loop mediated isothermal amplifications. So just a single temperature is able to detect the tuberculosis, and you don't need for any sophisticated image analysis systems. Because during the PCR, there is a pyrophosphate ions, and if it generates during the amplification, we could be able to detect from our naked eyes. So what we are approaching here, to develop a nano chip where we have different channels, and in this channel we will make a single temperature reaction for tuberculosis amplification. And there is a 2 chip, one is PDMS and one is a glass chip. In glass chip we have a battery operated, so it will be a pocket device. So even using the battery you can maintain a temperature. And if you have this temperature, we are able to detect the tuberculosis in the latent phase. And I think this is very promising device. Once we have this device, it will be very easy for everyone, and even at the home, they can able to detect the disease, tuberculosis. So that's, I think, very soon we are going to finalize this project, and it would be available for common persons to detect the tuberculosis in coming next year. So this is about the theme that why we need to discuss this healthcare issue and how we can come up with some innovative ideas to improve our healthcare by having a close interdisciplinary approach. Okay. About our speakers, I think they are very potential. Next, we are going to have a brain talk with Dr. Renu. Dr. Renu is coming all the way from Japan. Then we have Dr. Renu Prasad there. He is coming from JNU, another brain lecture. Then we have Dr. Yamanaka. Yamanaka is working on motor neuron disease. This is very, I think, trendy in next week. He is also coming all the way from Japan. Then we have Dr. Tanisana. He is coming from CAI. We are talking about the health disease. Then we have Dr. Kari is there. This is coming from Japan. She is also working on some protein misfolding, because all the diseases in the brain are associated with the protein misfolding. So how her work can be helpful to improve the brain disorder. Then in next session we have new technology in molecular medicines. Dr. Taka is there. She is also coming from Tokyo all the way, and she is developing some new biomaterials. I think it is very promising to jump on the new health devices. Then we have Dr. Sunil is there. She is also very senior person coming all the way from Japan, and he will be talking on mechanism of stress and some imaging, some new device imaging device that can help to detect. Then we, unfortunately, Dr. Madhu will be not here because of some reasons he was not able to fly. Then we have Dr. C in another session about this. Dr. C is coming from Vanastari. He will be talking about some precautions, how we can control the obesity. Then we have Dr. Sakata is there. He is also coming from Japan. Dr. Sakata will be talking about some new research on brain can be helpful diabetes. Then we have Dr. Munjal is here. Dr. Munjal is talking about some some of our resources, plant resources that can help to control our obesity problems. Then we have Dr. Bharti Malhotra from S. Medical College. She is a metabologist and working on tuberculosis. So we have collaboration. So I think it's also good to get Dr. Bharti here. And then we have Dr. Ashima. She is coming from IIR Gandhinagar, and she will be talking on some viral infections and tuberculosis. And then our final speaker will be Dr. Ashish Verma, coming from IIT Noida, and he will be talking on some HIV and his work on some HIV protein on this HIV. So this is all the speakers, and out of 16 speakers, 11 are from Japan, and 9 are from Japan, and only 1 is there from Europe. So all the speakers are coming from different cities in India and Japan, and I think it's the best time for all of you to share this knowledge. Okay. And Dr. Katus will be here tomorrow in the ceremony. I think it's a very good time to meet Dr. Katus also. And this time, because this is my particular interest to give a chance to our young minds to come over there and to share their research. And this time we got 72 abstracts, and out of 2 we have selected 10 young researchers. 2 is coming from Japan. Dr. Takehara doing something very brain device research, very important research, and Mr. Kamas is there, and another 8 we have from Rajasthan here. And also we have the workshop, and I'm sorry because it is limited, but we have 52 participants, and in this workshop we will have 2 different sessions where we will talk about microfabrication and tuberculosis. So let's sum up this. So every life, I think, is very precious and very valuable. So it's a crucial time when we should come together and to develop some new innovative ideas and new work which can be available to a common person to improve healthcare. So it's a very crucial time. Let's come be together and let's work on some interdisciplinary approach, and let's make some innovative ideas, and let's make this India or this world very safe, beautiful world. Thank you very much.
Sir, for your thought-provoking address to set the platform for our audience to deliver their thoughts in the area of healthcare and its importance. Now I would like to introduce our next speaker, Dr. Renu Wadhwa. She is head of Cell Proliferation Research Group, Biomedical Research Institute, National Institute of Advanced Industrial Science and Technology, Japan. A research interests are molecular mechanism of stress, aging and cancer. Her education is MPhil, PhD in Biology and Cellular Aging from Shukuba University, Japan. She has several publications in international and national journals. The title of her talk is Potentials of Coordinated Loss of Function, Screenings in Bioinformatics. Please, ma'am.
Thank you very much. Very good morning to everybody. I can see very enthusiastic faces sitting there. What I'd like to do is to first congratulate the Amis, 3 young brothers having the task of setting up this very innovative and with big dreams of having this institution going a long way. The kind of energy, enthusiasm and efforts I see putting in, I can already see it rising to excellence level. So congratulations to the Amis first, and then thank you for having us here. It's very nice being in Jaipur and to be able to interact with the young faces here. So what I think I'm going to do is to tell you guys what we have been doing in the last 20 years in Japan, trying to tell you how scientific questions and how with technology we can answer one by one questions and how this can develop into technology or application. So the title of my talk was Aging and Cancer, and I'll be touching on basic and interventional studies. Before doing that, I'll give you a little bit glimpse of the institute I come from. It's called AIST, Advanced Institute of Science and Technology. We are right from north of Japan, from Hokkaido to south in Kyushu, and we are here in the main AIST headquarters, a city called Tsukuba Science City, about an hour drive from Tokyo. In AIST, what we do is we deal right from molecules to machines. and that's a real integration. We do research on the molecular level, and we also make the machines. So that's a real platform for doing things together, integrated platform. So my group here called, I'm not sure whether you'll be able to read it here, Biomedical Research Institute, and we are called Cell Proliferation Research. As the term goes, cell proliferation, everybody who is not even doing anything with the biology can understand it is the proliferation or division of cells. And what we do is, by using basic and applied platforms, we try to understand what controls proliferation. And to tell you what is cancer, cancer is uncontrolled proliferation of cells. So if we understand what controls proliferation, there will be possibilities of controlling or treating cancer. Okay, let's deal with the questions of cell proliferation control, and what you will be stuck with thinking, what is aging first? We all know what aging is. It is a natural phenomenon, and of course it is a fear. And I think not many of you feel this fear because you are very young. You start feeling fear when you see your family members getting stuck into the aging troubles, and then the fear comes. So anyway, what I'd like to remind you here is, just like aging, what you see here, our cells, when you culture, if they are normal, they also age. So you can have young cells, middle-aged cells, and the old cells. So what we mean to say is that we can use these cells as very nice model to understand what aging is and what will prevent aging, or what happens when there is uncontrolled aging. So another thing I'd like to remind you is that as you age, you of course see increased incidence of many diseases, and cancer is one of them. So if you equate the animal model with the cell model, I'd like to remind you, I'd like to tell you here is that here, normal cells, they go through a phase called middle age phase or aging phase, and it is only the aging phase when they immortalize, they turn into cancerous. In other words, in vivo system as well, it's the aging and cancer very nicely linked. So in my group, what we are trying to do is, we take normal cells and cancer cells in parallel, and we ask questions. Why these normal cells, they can divide only for a limited number of times? On the other hand, why the cancer cells can go on forever? So taking these two as a basic model, we try to ask these questions. The first question that we asked back in 1990 when I started as a postdoc in Japan was that, is it a loss of function or is it a gain of function? So to answer that question, what you would do is, you will cross the normal cells with a cancer cell, and you will get a hybrid cell, and you study if this hybrid cell is cancerous or normal. And what we found was, this hybrid cell is modal or normal kind of phenotype, which means that aging phenotype is dominant over the cancerous or immortal phenotype. Fair enough. So that's not really enough. What we need to find out is, what is there that is controlling immortality or mortality over immortality. So the approach we had taken was just to compare these hybrid cells, the parent normal and immortal or cancer cells, on a protein gel like this. And we were lucky enough to find one protein, you see that as a form of a band here, that this protein was very much coming with the mortal cells, but absent in the immortal or the cancer cell. So there we named it as mortal. So then we know that this is something that is, inherited with a mortal phenotype, but then what it is, having just single protein in hand is not really enough. So what do you do next? So what we did was, we have this protein purified from the gel, and then the next step is you raise an antibody. You can use different animal models for that, and we use rabbit to get this polyclonal antibody. And once you have that in hand, you see have you been lucky enough to get an antibody that will see your protein? And we were very lucky that way. Here is the game. We see that this antibody recognizes the same protein that we had first identified on the gel. Okay, so now we have the protein, we have the antibody, we have to move on. We want to know what this protein is, we want to identify this, and which means that you need to know what is the sequence of your gene, or in other words, what is the mRNA code for this protein. So here we have cloned it by a approach called expression screening or expression cloning. What you do is you express all the human cDNAs into a bacteria, express them on a plate like this, and then you use your antibody to recognize your specific clones. And by that way, you have a sequence of cDNA that will translate your protein. But you really need to make sure that the sequence that you have is really your protein or not. So once again, you in vitro make your protein from the cDNA that you have got, and then you do immunoprecipitation, which means that you're using your antibody to pull down your protein. And if that works, that means you know what your protein is, and then you can go ahead, you can sequence, and you can actually read your protein in form of a DNA sequence. And once you know that, you can get into the bioinformatics, put it into the database, and see what your protein was. And we having done all this process, we find out that our protein was a new member of an HSP70 family of stress proteins. So there, by nearly 3 years of work, we had cloned a protein which is a new member of HSP70, and we had named it as Morgan. Okay, so now we've got this protein. If you remember, I told you that it is coming up with a model phenotype, and then it is a new member of HSP70. Since it is a heat shock protein, we need to know whether this is inducible or not. And if this is a gel where you're looking at the protein, this is a sample which is heat shock, you can appreciate that this particular protein is not heat shock induced. What other proteins are usually, the heat shock family proteins are induced if you heat shock yourself. But this protein, which is unique, it is not heat shock inducible. And another thing I want you to remember here, all these other heat shock proteins you see by arrows there, they are not seen by the antibody that we have raised. In other words, the antibody we have is extremely specific. And then we've got the cDNA, we know this is a protein, we know that this is coming with a model phenotype, we want to know the next functions, what biologically this protein will do. So for that, you need technologies like you should be able to express your protein into the cells, you call it overexpression, and then you see what it does to your cells, and then from cells you can move on to the animal system. Here we express the protein, but what we found in the mouse cells, it makes them more malignant. In other words, that it is making the cancer cells more aggressive. The ones which never form tumors in mice, they start making tumors there. So what we understood as a first step was that this is the protein that is helping cells becoming cancerous. So that's a nice phenotype, and then we need to work on the mechanism. So here, I'd like to tell you another protein which is called p53, and it is the guardian of our cells or body, protects us from the DNA damage. So what we found was that our protein modeling and p53, they exist together in cells. So this is the double immunostaining where you see yellow color if the two proteins only co-localize. Otherwise you will see either green or red. So this is the co-localization between p53 and modeling. These two proteins are existing together, or they are together in the cell. And then if you, in an assay where you are looking at the activity of p53, if you have more modeling, you will actually bring down the activity of p53. Then we did an experiment. We overexpressed modeling into the cells, and we found that it is actually knocking down or inactivating p53. So as a model, it came up a few years ago that p53, which is a nuclear protein, this needs to go to the nucleus, but it is kept in the cytoplasm by binding to the modeling. So that is the way it is actually inactivating p53. So as I introduce p53 protein to you, this is a transcription factor. This is a transcriptional factor here. It takes care that the cells does not divide uncontrollably. So it takes care of DNA repair, and when there is a damage, the cells either arrest or they may get into apoptosis. But if p53 is inactive, cells keep on dividing, and that's what is cancer. And I'll tell you that about 80 to 90% of the cancers, they have mutant p53, all the p53 activity is lost. So the interactions of modeling to p53 does make sense that it is promoting carcinogenesis by inactivating p53. So the next question comes, okay, if overexpression of modeling is making the cells cancerous, what next? Can the modeling loss or compromise of modeling can help in cancer therapeutics? Okay, if we did that, you can actually knock down modeling by a number of ways. Here we are using antisense, and when we put that antisense into the cells here, they are growth arrested. The another way to do the same experiment is to use ribozymes. If you use ribozyme, you bring down modeling level of expression in the cells, and the cells again, they are arrested, not dividing anymore. So most recent technology that has developed in last decade, the siRNA. So if you use siRNA against modeling is knocked down. This red staining means movement expression. You can see that movement is knocked down, and these guys here, these cells, they are not dividing, they are actually undergoing growth arrest or apoptosis. So the model becomes like this. That normally p53 and modeling complexes which are in the cytoplasm, and that way p53 is inactive. What we were able to show was that if we knock down modeling like this, p53 is able to get back to the nucleus, and that can be activated. We also showed the same phenomena happening with the chemicals, and we also used the peptides, again bringing p53 back into the nucleus. And the result of all this was putting cancer cells into a growth arrest situation. So what I have told you is the protein that we cloned, modeling, if we can manipulate its expression, overexpression makes the cancers, and if we make it suppression, we can actually suppress the cancers like this. So for all this, the modeling story which has developed in last 20 years, it summarizes into two sentences that modeling promotes human carcinogenesis, and anti-modeling molecules, they are potential anti-cancer reagents. Alright, so that's working with one protein. So you probably have realized that to know functions of a protein is biological significance. You have to go through all the technical platforms. You should be able to do all these questions, ask these questions and answer them with the recent technology. Now this understanding of basic biology of the protein that goes on, and it takes about years and years and billions of dollars to develop these outcomes into some medicine. We thought, while we are understanding that, we need to have some intervention which is easily possible. So intervention, by intervention, what do you mean? So some of us will prefer prevention, of course. Some of us will go to the drugstore and say, okay, I'll have an ounce of prevention, and others would prefer the drug. So we thought that something which is already there, we have it every day. For example, we come across Ayurveda principles and we see in our home, in our kitchen, Ayurveda is being used. We know that the traditional Indian medicine has a history of 5,000 years, and our mother, our grandmothers, they use it for any kind of thing, right from the headache to cough to sore, and most recently we have it used for the beauty reasons as well. So that holistic thing that's there already in the understanding in our minds, why hasn't it gone to the laboratory and come out as a polished form, easy form to absorb? So we picked up a plant called ashwagandha. I'm sure you all are aware of this name. Why ashwagandha? Because it's got a long history of health benefits for various kind of reasons. It's been used in our Indian home medicine. And then what we started with was, this is only a 6-year-old project, and what we started was start making the ashwagandha extracts, and folic extracts and the water extracts, and then put those extracts into our assay systems. And here you see there's a cancer, there's a premature aging, there's a toxicity by industrial metabolites and neurogenesis. And we start testing the activity of these extracts and the components in the cell culture model and the animal model. One of the first experiments, because of our own interest, was done using cancer model, and here you use the nude mouse, and what we found was the ashwagandha and folic extract, I call it as an eye extract, is having a very strong tumor suppressor activity. Both by as you give it as a local injections into the tumors, or if you give it by overfeeding. But at one stage, it was a very frustrating project because we see the activity, which is the tumor suppressor activity, is very variable. Some of the batches of extracts that got very nice activity, but the others were too bad. So if you got results in the lab like this, you're totally frustrated. But then we were lucky enough to have chemotypes and the activity always matched. So we very quickly figured out that the tumor suppressor activity was matching with this peak, this chemical, this phytochemical in ashwagandha alcoholic extract was the active anti-cancer component. So we had purified it, characterized it by chemically by NMR analysis, and we know that this is limonol. Now, we've got ashwagandha phytochemical limonol, which is showing an anti-cancer activity in our assays, but then the mechanism, our main purpose here in the lab is to find out the mechanism, and we took again our favorite normal cells in the cancer cell system. What I'd like to show you here is that eye extract is killing the cancer cells, but it is safe for normal cells. Similarly, limonol is killing the cancer cell on the top, the orange bars, but it is safe for normal cells. In other words, it is a selective toxicity for the cancer cells, which is very much wanting. So here also you see the cancer cells on the top, they are getting killed by ashwagandha eye extract and limonol, but the other 3 kinds of normal cells, they are happily dividing and they have no effect. So what we found was that eye extract and limonol kill the cancer cells selectively. Once again, you have solved one question, you have raised another 6. So here the question is how? Now here the technology that we have used was siRNA screening or loss of function screening. So what you do is on a level 96 well plate, you plate the cells and then you knock out one gene by one gene in one well and see the effect that you desire to see is lost or not. And if it is lost, then that particular gene is the cellular target, or that's the gene that is involved in action of ashwagandha like this. For example, I'm talking about selective toxicity. So we took this platform and we identified many genes, and out of these, the ones labeled in yellow are the prominent ones, and we found that P53 is again there. So tumor suppressor P53. Then we took an alternate approach. We drew a pathways there which will be involved based on the cellular targets that we isolated. And again we found that P53 and BCL2 were there. Then we took an unbiased approach where we knocked out not gene by gene but by a pooled ribozyme library. And then the cells that survived, we rescue the ribozymes from there and we see that what are they and what are they knocking down. And once again, we draw by the help of bioinformatics the pathway like this, and it comes right. One is the P53 pathway, the other is the reactive oxygen species pathway. So in other words, the oxidative stress and the tumor suppressor activity. So I showed you by the 3 independent approaches. What we isolated was the information that P53 P21 pathway as a candidate target, and the other one by the approach 3 was the oxidative pathway. So let's now validate whether this is true or not. Is P53 selectively activated in cancer cells? Let's see here. What you see on the left-hand side is the cancer cells, and this is the normal cell. These are the control, and these are the early extract or the non-treated cells. So you see this band has gone up as compared to here. This means P53 is activated by treatment of cancer cells with either the ashwagandha alcoholic extract or its component Vidanone. But you see, there's not much difference in the normal cells. So that validated that what we have isolated to begin with is true. So looking at the mechanism again, we saw that if Vidanone actually docks into P53 or not, and you see the model of P53 as a protein as blue, and you see that little thing that is Vidanone, actually it can hook to the pocket of P53. That seems to be very much important, and we had shown that the same region is the one that seems to be involved in binding of P53, the chemicals that activated P53 in response to Volcan as well. So the other protein that is involved in regulation of P53 is MDM2. On the left-hand side, you see P53 and MDM2 complex, which is inactivated. And here, the little thing here, the again green, it's Vidanone, and it's taking the place of MDM2. What it means is like this. So once again, just remember the model. More than P53 in the cytoplasm, which is inactive, and here we are talking about Vidanone. What P53 needs to do is to go into the nucleus, but in this case, what happens is, in spite of going to the nucleus, it hooks into Volcan or MDM2, which means the P53 is inactive. So when Vidanone is there, it releases P53 in Volcan and MDM2 complexes, making it active as it did in case of when we hook down Volcan, in case of ribozymes and siRNA, and the result was gained in the rest. So Vidanone, in other words, hooking to P53 and MDM2 will activate your tumor suppressor pathway. So that we validated. The second thing was the oxidative damage. The best thing to look at oxidative damage here is the mitochondria. I am not sure if the screen is so clear or not. The mitochondria are ruptured when you treat the cancer cells with Vidanone. And what it means is that the effect of extract and Vidanone is selectively on cancer cells because of the oxidative stress as well. We also looked that Vidanone is having what kind of effect on the normal cells. So we cultured the normal cells in Vidanone supplemented medium, and what happens is the cells have the extended lifespan. What in other words it means extended lifespan of normal cells is that you will have few more candles on your birthday cake if the lifespan extended. That's not only enough. You want to have a healthy lifespan, which means that you want to look into the oxidative damage. That's what we did, and you can appreciate here the Vidanone fed cells, they have less oxidative damage or less cross-linked proteins, which means that the Vidanone is protecting the cells against oxidative damage. Not one kind of oxidative damage, many kinds. And here's the UV stress. This is the UV stress in the normal medium, and this is UV when you have eye extracted. And you can see that more number of cells in this panel as compared to regular ones. And then another kind of oxidative stress you can have the hydrogen peroxide. Once again, you can see that the Vidanone is increasing the viability of cells there. So the very important question in the present world that we are living in is the industrial chemicals. So we also use the industrial chemical stress, and here we are using MAA as a chemical, which is a model for the industrial metabolites. And you see the cells treated with MAA here, they are dying out, but here in this lane, they are co-treated with Vidanone. So they are doing much better as compared to these. So MA-treated cells, which are chemically intoxicated, they are undergoing senescence or premature senescence, and they are recovering well if Vidanone is there along with MMA. So that's probably a summary of what we did on MAA. So there is a more senescent cells, you see them as a blue color. There is more DNA damage, you see that as a green fluorescent stain, and there is a more ROS damage there if you have the cells treated with MA. But if you have MAA and methanol, you don't see all this damage happening in the cell. So in other words, we don't know normal cells and it is killing cancer cells selectively. And is it really selective? We see in comparison the normal cells and the cancer cells. This green staining tells you that they are damaged. You can appreciate that all these cancer cells are damaged, but these normal cells, they are not undergoing any damage if they are treated with eye extract or methanol. So that's the safety of methanol. In other words, another way, cancer cells and the normal cells, the cancer cells are damaged by the reactive oxygen species, but not the normal cells. So by this, we can keep on doing the technical experiments, and what we have found out is here that ashwagandha extract, which is a folic extract, and its component methanol, that is selectively killing cancer cells. The activity belongs to methanol, and the mechanism is the activation of p53 tumor suppressor pathway and the selective oxidative stress to the cancer cell. So I'd like to stop here and acknowledge the institute. AIST is where I come from, and this work has been done in collaboration with other institutions, including Guru Nanak University in Amritsar and IIT Delhi. And I'd like to also acknowledge people, my students, who have actually done the group work. The top group is Modern Group, and this small group is actually the group that done the work that I presented. Thank you very much.
Work. And one is very encouraging to see that your extract has a selective killing of cancer cells instead of normal cells, because one of the drawbacks of any treatment is selection is not there. It is easy to kill a cancer cell tumor, but the major aim is to protect the normal cells. That is what is the treatment. So it is an excellent piece of work, and I hope that ashwagandha will have a good future. I just want to ask you one or two. I was thinking around that if you give ashwagandha to human beings or patients in future, what should be the mode, oral mode or is any injection mode, number one? Whatever may be the mode of injection, will there be a selective absorption of this extract to the tumor cells only, or normally it will be distributed and then it will have effect? Because and the second question is, even if it is a selective absorption within the tumor, and we know that in the tumor there is an oxygen gradient, so you have oxic cells, hypoxic and anoxic. So the major problem of any treatment is this anoxic portion. You can kill this oxic portion of the cancer cells, but these anoxic cells remains unaffected and later on it grows. So my point of view is how you can comment upon this.
Thank you very much for the excellent question. The first question is, what will be the mode of administration if we get to a stage when we are using it on human beings? My answer would be, my preferred mode of administration would be the oral. And we know that ashwagandha has been in our kitchen, in the traditional medicine for years and years. So there's no reason to avoid why can't we eat it unless we can rule out the toxicity. And the findings that we have come up with, the selective toxicity to selective killing of cancer cells is very encouraging. So the second question, whether it will be selectively absorbed by cancer cells or not, we are not sure at the moment. We're trying to do it. It's a kind of difficult experiment because it's very difficult to track after you have treated the cell. You see the phenotype, you see the effect, you're working on the mechanism. But it's very difficult to actually see whether how much of the unknown has gone into the cell. We are trying to develop antibodies to these small molecules, and we are also trying to tag these purified chemicals so that we can actually see that what selectivity you see in terms of killing is is actually in terms of absorption or it is the mechanism beyond that. So that's something we are doing. And the third question was the oxygen. When you look at tumors, there are different parts of the tumor. The center of the tumor maybe is growing very, very dense, so it is hypoxia situation. So how is it going to affect if the mechanism is the oxidative stress? So I think this in other words also translates into a question whether you can have different aggressivity of the tumors treated by sugar and stress. So again, today I can't really answer you for sure, but this is definitely our plan of action.
I think it is very selective for the cancer.
Sorry,
Why it is selective for the cancer? Because it's a common metabolite, a product present in the ashwagandha. Why? Yes.
We don't know whether they are any cancer cells, they are different. For example, they are different in the mitochondria membrane. They have different cell surface antigen. So if this component is giving you the different phenotype because of the different mechanisms, or just because the cancer cells are more adaptive to these.
The second question is that most of the time you can use chemotherapy or other means to control the cancer. Cancer you can't cure. But the problem really comes when the cancer patient has a remission, and that happens because of the cancer stem cells. In that case, this is very effective. Have you ever tried that?
Yes, it works very well for cancer stem cells. We have published that, but we doing that.
Thank you.
It works excellent on the cancer stem cells.
So maybe 1 or 2 minutes.
Mentioned in your slide that in a cancer cell, these different molecules co-localize. So have we any study on what causes that process to initiate?
What causes?
Do we have any study on what causes that process to start? Any environmental factors or?
Well, the first thing is that most of the cancers, hormone is upregulated. So as the cancer develops, this is a stress protein, so it responds to the cancer situation, and most of the cancer cells are enriched with hormone. So one thought is, okay, it is now overexpressed, now it is functioning to keep the p53 under control, and by that, it is contributing to cancer.
Any other data that how this component of high stress has actually increasing?
The kinetics of binding have not been worked out, but the immunoprecipitation is a standard way of looking at the binding. We know that it disturbs the binding of p53 hormone complexes, but the efficiency or the kinetics we don't know yet.
So let's make a thanks to Dr. Renu
Thank you very much.
for giving us the